Welcome to Justice Today, the official podcast of the Department of Justice's Office of Justice Programs, where we shine a light on cutting-edge research and practices and offer an in-depth look at what we're doing to meet the biggest public safety challenges of our time. Join us as we explore how funding, science, and technology help us achieve strong communities.
Hello, I'm Jim Dawson, the Senior Science Writer at NIJ. I'm speaking today with Greg Dutton, a Forensic Science R&D Program Manager at NIJ, about the microbiome. Greg's portfolio includes trace evidence, impression and pattern evidence, and the microbiome research being done by NIJ. Over the past decade, the human microbiome, consisting of trillions of bacteria, fungi, and viruses that live in and on every one of us, has become increasingly important to criminal investigators as they've realized that we all leave traces of our unique microbial evidence wherever we go. The field of microbial forensics was born out of necessity in response to the 2001 anthrax attacks in New York, Washington, D.C., and Florida that killed 5 and infected 17 others. The research in microbial forensics has advanced significantly since then. Greg Dutton has been working with the microbiome researchers funded by NIJ. So welcome, Greg.
Thanks, Jim. I'm really looking forward to talking about microbiome today. I should note that this is just a small portion of NIJ's forensic science R&D, but I think it's a really interesting one.
Let's start with the basics. Can you describe the microbiome? What is it? Is it unique to me? That kind of thing.
Yeah, so microbiome, if you think about the word biome, biome is a community of living organisms, right? Microbiome is a community of microscopic living organisms. So we're talking about things like bacteria, viruses, fungi, right? These are the things that live on us and with us. We carry bacterial communities in and on our bodies. So our gastrointestinal tract has a microbiome that's very important to our health. Humans have a microbiome, a community of microorganisms on our skin that actually helps to keep our skin healthy too. So these communities of bacteria, viruses, they're adapted to the environments that they live in, right? So some of those are the environments of our body. So they're adapted to the environment of our skin. And, our skin is different between individuals, right? So, some people have drier skin, oilier skin. So the microbiome that we carry depends, it's characteristic of an individual, but it's not necessarily identifiable to an individual. So I think that's the distinction there. We're talking about communities of bacteria that are characteristic of everything about us: age, sex, diet, health, but not necessarily something that can be tied to a specific individual person.
Okay, thanks. And I understand, again, we're talking huge numbers, right? Each of us has trillions of bacteria on and in us. So this is a major community that we carry around with us that we just don't see and are often unaware of. Right?
Yeah, I know, you've probably seen it said that, the majority of our body is really not human, right? It's made up of bacteria. I don't know to what extent, that's true, but definitely there are trillions and trillions of bacteria and viruses that are a part of us. They adapt to us. They're characteristic of us. They tend to have some stability over time. So if you sample the microbiome of an individual over time, it will tend to have some stability, and it will have some differences from others. Another interesting thing is that, it's been shown that the microbiome of people who live in the same household tends to converge over time to become more similar. Also, people who have pets, they pick up bacterial populations from their pets that then adapts to live on and with them. So there are things about the microbiome that are characteristic and telling about us.
I could scare myself thinking about this, but for the most part, this is a good thing, right? They're there for a reason. They do us good when things are
absolutely
functioning normally. Yeah.
Yeah, for the most part, right. We're living in equilibrium with them, right? And there are, some that are really critical for our health. So a lot of the research, that we're talking about in terms of forensic trace microbiome, the research really was started in health and medicine research because it has been increasingly understood that our microbiome is critically interrelated with our health and wellness. So that was the spark for this whole research area. And now researchers are just seeing how it can be applied to forensic problems.
Greg, I understand that advances in DNA science and computing over the past decade have increased the likelihood the microbiome can become a useful tool in forensics. What exactly has changed from 10 years ago or so that makes this possible now that wasn't possible back then?
Yes, so there are a few things that have really come together. The first thing was the development of the sequencing technology. So this really came out of the Human Genome Project in the late 90s. So that set the stage. So researchers had the tools to sequence the microbiome. And the next thing was really figuring out how to make sense of all the information that they were getting from this sequence data. And that really came together with under the Human Microbiome Project that followed on in the mid to late 2000s. And the key thing that happened there was the microbiome community really converged on a standard for analyzing and comparing the microbiome. And it's called the 16S ribosomal RNA profile standard. We can talk about sort of specifically later, what that is. But the key thing was that they all had kind of a standard to use to measure and define how similar or how different, a microbiome was to another. So those are the key things that came together. Now, with that, then they were able to go on and, apply these analytical techniques to, certain areas, and forensic science is one of them.
So when did NIJ become interested in this? I know we support a lot of different research in different areas of forensics, but microbiome, how do we get started on that?
So our support for microbiome research has grown over the years. I think one of the first forensic areas that microbiome was used to analyze was actually analyzing soil evidence, right? So that's a little different, right? Certainly different from, human skin microbiome, but, soils have characteristic microbial profiles for sure. That was the first area. Then, after the Human Microbiome Project, the researchers under that were looking for places that they could apply their expertise. And so, I think in the early 2010s, we started to see those researchers come to us with ideas for how they could use their microbiome techniques to try to answer forensic questions. And so we started to see proposals looking at, I think the first ones actually were in decomposition. So using microbiome as a tool to follow the decomposition process to try to determine how long it's been since, a cadaver has been decomposing. Some of the researchers that started that, they were at the University of Colorado. They moved to UC San Diego. They then got interested in the area that we're talking about now, which is sort of skin microbiome and touch transfer of the skin microbiome. and we funded a number of projects under that in recent years. But there are other areas of forensic application that microbiome is active in now. Body fluid ID, for example, if you have a stain that's collected from a crime scene and you want to figure out, what is that? Is it blood, semen, saliva? Microbiome is a way you can help classify that. So we've seen it touch all kinds of corners of forensic science.
And in the touch side of this, I understand, looking at a couple research reports, that there's sort of a last person touching. So if you have 4 or 5 people in a room, or if, I think in one of the studies, they had fake burglars come into a space and touch objects that have been touched by the residents, then they were kind of overlapping and they couldn't distinguish earlier touches. So I guess, how good is this? What does it really tell you at a crime scene when you swab surfaces and find samples? What are you really learning from this?
So that's an interesting project. So this is a study from University of Chicago, Jack Gilbert, and that's interesting because they were building on some prior studies that sort of established the fundamental questions. So they first had to study and determine whether a person's skin microbiome is stable over time, right? Does it change? Mm-hmm. Can it be differentiated from someone else? So they had to do those things first, right? Then they went to more sort of classically forensic questions. So questions there, so this mock crime scene that they did, the questions are, is evidence transferred from a person to a surface at a crime scene? And can you collect that and measure it, right? So I agree, it was interesting. They had some success, but they were using a fairly small set of people. So, it wasn't exactly like a real world scenario. And the most important thing that they found out that actually is going to be a challenge for using microbiome as trace evidence in the future is they found that they had to collect the evidence within an hour of it being deposited. So, one of the things about, our skin microbiome, it lives on us, but it needs our skin. Like, that's the best environment for it, right? When it's transferred to a surface, it's dying, right? So if you don't get to it and collect it and keep it cold pretty quickly, it's going to change. The characteristic profile from the person who deposited it will be decaying, right? So that's one challenge is, you need to collect it quickly. You said, they noted that it tended to retain the information of the last person touching, right? That also is true, although that may not be so bad from a forensic perspective. If at least you can get the last person touching and you predominantly see that, you may be avoiding getting mixtures, which is a real challenge and problem with human DNA evidence. So that was an important proof of concept study, but it definitely showed some challenges that forensic microbiome for touch analysis is going to see in the future.
Thanks for listening to part 1 of my conversation with Greg Dutton, and tune in to part 2 for the rest of our discussion of the microbiome.
To learn more about today's topic or about NIJ, visit the links in the episode description and join us for new episodes every month. Opinions or points of view expressed in this episode represent a consensus of the authors and do not necessarily represent the official position or policies of the U.S. Department of Justice. Any products and manufacturers discussed in this episode are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice.